Okay, I'm going to give you a little background information on Cambridge, a little bit about the regulatory and science involved with autogenous and why we believe autogenous is going to be a growing need going forward. In the upper left-hand corner up here, you see our manufacturing site in Sioux Center. That's about a 37,000 square foot facility with multiple air handling sites on it, so it's like several buildings under one roof. To the right here is our administration office. That's where our administration, sales and marketing, customer service, account management is located in Worthington. And down here in the lower left is our diagnostic lab and our research lab. So those are in Worthington. Wayne and I have been asked several times why we're doing this. And our answer to that is we believe that there's no greater need or opportunity today. We have new technologies that are greater than ever. When I started in this industry, we knew very little as far as what was the genetic information with a particular pathogen. Today we can do a complete genome sequence in one day. We also have emerging and re-emerging diseases, some of them associated with the Veterinary Food Directive. We'll talk about that in a minute. You're familiar, I believe, with antigenic shift and drift. And the other point here why we think it's important to be doing this now is the need for fast and flexible responses. With an autogenous vaccine, we can have that to the veterinarian in 2 to 4 months, where it takes 4 to 6 years to develop a commercial vaccine. The other is the changes in regulations and costs of doing things today. When I started in the industry, we could fully license a particular vaccine in about 2 years for $500,000. That same technology today would take 4 to 6 years and multiple $1 million. So return on investment isn't always there. So we believe, again, that there's going to be a growing need for this technology. So what are some of the other opportunities for autogenous vaccines? Pathogens with no commercial vaccine, such as Group C rotavirus, which also doesn't grow in cell culture, so looking for other ways to do that. There's a variety of emergent diseases that we're seeing. Swine neurological disease, atypical porcine pestivirus, passivirus, cephalovirus, teshovirus, and enterovirus, just to name a few. Also the genetic variable pathogens where commercial vaccines don't always work. Swine influenza, which you're very familiar with, and porcine reproductive and respiratory syndrome virus, which you're also very familiar with. Add that to the fact that we can put different combinations of vaccines together to save cost and labor. The changes in the antibiotic usage, we believe we're already seeing some of the reoccurrence of some of the diseases that we haven't seen for a long time. It's put an increased emphasis on prevention versus treatment. Again, we licensed a Streptococcus suis vaccine some years ago, there were basically 3 types in it. We had a type 1, type 2, and a type 1 cross 2 in it. Today we have 35 plus serotypes that those commercial vaccines won't touch. Same thing with erysipelathrix. When we licensed that vaccine some years ago, it had 2 serotypes in it. Today there are over 28 different serotypes of erysipelathrix that the veterinarian and his or her client have to deal with. This is a recent publication. I won't take you through it, but it was a study that was done on 6 Midwest swine operations. Just dropping down to the bottom down here, it showed that the commercial vaccines worked on 1 of them, but it said however, vaccinal and clinical isolates were of heterologous serotypes at the remaining 5 sites, suggesting that reevaluation of vaccine efficacy using recent field isolates may be warranted. From a regulatory standpoint, autogenous vaccines fall under 9 CFR 113113. The CFR is Code of Federal Regulations. Their standards for manufacturing, testing, safety are very similar to what I call them, the off-the-shelf or the commercial vaccines. We have to have filed and approved outlines of productions. We have to have filed and approved standard operating procedures. Our QA is very similar. With the adjuvants that we use, we have to do withdrawal studies and safety studies very similar to the non-autogenous type vaccines. Same with our cell lines for doing viruses. We have to have USD confirmatory testing after we've looked at adventitious agents and karyology. That all has to, it's all very similar to the non-autogenous vaccines. Same with master seeds in the autogenous. We can use those seeds for about 24 months. Expiration date on the product is 18 months. There's non-adjacent uses. I'll cover that in a few minutes, and we'll touch a little bit on the prescription biologics. It all starts with diagnostics. And these are the some of the services we offer: bacteriology, aerobic, anaerobic, antibiotic sensitivity, serotype, pretty standard stuff you're all familiar with. We do virus isolation and identification. But the very interesting thing and positive thing that we have today that we didn't have for years is this molecular diagnostics, PCR and the sequencing that goes along with that. In vaccine development, these things are very similar to standard or custom vaccine. We have to know what strain to select. We have to know how to apply it. We have to know what genes that are important in the pathogenicity and how to express those genes, how to formulate. Looking at efficacy and potency. This is the main difference between autogenous and non-autogenous. Efficacy is not something that you prove in a non-autogenous, nor can you claim, but we build it in, and I'll talk about that in a minute. Field safety is very similar, as is quality control. As far as strain selection, this is just one example. Again, taking E. coli with the pili adhesin. Again, years ago, we licensed a product that just had K88 and K99, F4, F5. Today, we have a number of other adhesins. We used to have on the E. coli toxins, when we started in this, we had the STA and the LT. Now you can see on the list here of all the other toxins that we have to be looking for and working with. We also have other adherence factors as listed here. With Clostridium perfringens, we used to just focus on the beta toxin. Today we know that the beta-2 toxin is very important, as is our other toxins such as the epsilon. This is an example of a Clostridium perfringens. These first 3 strains here would come from the field. The first 4 strains from the field. The 5th strain here is the positive control, and here's the negative control. All of them have alpha toxin, which you'd expect with Clostridium perfringens, but you see the beta-2, which is also very important. As we look across here, some of them don't have the beta-2. This is the positive control again. This would be the strain we would select for the master seed. Moving forward, we have DNA sequencing, so we can not only just identify the pathogens, but we can do a genetic comparison to the historical or regional strains and target on virulence factors. Ben's going to touch on metagenomics, so I'm not going to mention that. Once we have picked the right one and we know what genes to look for as we're producing the vaccine, we have to make sure that those genes are expressed. And so this is just an example where we have a control here and our control here, and then the sample where we get positive expression. As far as formulation, what we use is we've been involved in fully licensing a number of vaccines through the years, and we've established what's called MPDs, minimum protective doses. This is just a small sample of the MPDs for vaccines that we've licensed through the years. And we use that information to formulate our autogenous vaccines. Even though we can't claim efficacy, we want to make sure that the potency standards are equal to or better than what would be needed in a commercial vaccine. Same thing, we have a variety of adjuvants that we can select, and we also have to consider age in the development of our autogenous vaccines. Quality control is a very important aspect. As I'm sure you all know, we want to ensure the highest level of quality for autogenous vaccines. We need to verify the titers I've already mentioned, identify in the production harvests. Ways that we do that, some of the ways that we do that is real-time PCR for SIVs, same with PRRS. We also speciate and do real-time PCR for Mycoplasma pneumonia. This is just an example of the real-time PCR. We have standards that we have set up, and then this here line right here is the unknown. As we do the correlation coefficient, you'll see that the unknown has just a little over 8 logs. So on that harvest, we'd know that we'd have to concentrate that to get to the potency level that we need to have the product that we want to put out there. Continuing on, subject to the same quality control standards, subject to the same 9 CFR sterility and lab animal safety testing as are the off-the-shelf vaccines. We have internally mandated, as I've already said, harvest criteria, whether it's CFUs per mil, TCID 50, qPCR, as I've mentioned, and so on, to be able to build that in, although we don't claim potency again. 9 CFR autogenous vaccines are inactivated. Again, we have a variety of adjuvants to bolster immune response, from humoral to mucosal to cell-mediated. One of the exciting things that we are working with is a platform technology where we're able to, there's a USDA memorandum on that, where we're able to take a gene from another pathogen, insert it in here, and have basically what amounts to a subunit autogenous vaccine. That's something we're working on today. We don't have that USDA approved as yet. As far as usage, autogenous vaccines originally just for the farm when it was isolated, but now with the regulations, we can have non-adjacent farms with an epidemiological link where the animals move across country. We've actually had autogenous vaccines that have been approved across multiple states with multiple veterinarians involved in it. Also, another thing here is where we can deal with just regional vaccines dealing with a particular virus or that's in a given region and focus on that. This again is our USDA licensed facility down in Sioux Center, Iowa. It's about 50 minutes south of Worthington. The equipment that we have, again, is very similar to what you'd find in any of the commercial vaccine companies. This is our fermentation lab. This is our antigen concentration facility. We have a auxiliary lab that we call it, or we can do things like adjuvant preparation, media prep, stuff like that. This here is our autoclave and sterilization area in there. We have a batching tank here that goes our maximum cereal size would be about 1,500 liters. We go down to much smaller, and Stephanie and Mike can comment on our minimum dose size. Then we have a large sterile fill room. You see up here HEPA filters, so that's a very clean area, very similar to what you'd find in a commercial vaccine company. So in summary, autogenous vaccines are produced very similar standards to commercial vaccines. Outbreak to bottle, this is very important, 2+ months, 2 to 4 months versus the 4+ years it takes for commercial vaccines. And again, a lot of the things we're dealing with, these emergent viruses, may just be transient, so you need to deal with them, but there wouldn't be a commercial vaccine. We can customize these antigen formulations to a particular system, to a need. We have the ability to address diseases where commercial vaccines efficacy is poor or not available, and the ability to combine antigens, make a combination vaccine. Again, using our commercial vaccine development, we look at things like antigenic interference, so we know how to formulate these things to give us the best possible response. So with that, I thank you and open to any questions.